Photosynthesis, as we all know, is the unique process by which green plants prepare their food. Without photosynthesis, life on Earth will vanish in about 25 years. But do you know how photosynthesis and photosynthetic organisms evolved? Do you know the difference between oxygenic and anoxygenic photosynthesis? Do you know the location of photosynthetic pigments? Let's discuss in detail. Hello everyone, I'm Shanty and you are watching Biology Nowadays. When life began on Earth, the first organisms that came to Earth were heterotrophs. The term hetero means other and troph means one that feeds. Heterotrophs being organisms that feed on others. In other words, heterotrophs didn't have the ability to prepare their own food. So what did they do? These organisms depended on the amino acids and proteins that were present on Earth at that time. As a result, these organic compounds started to get depleted. Perhaps the first famine on Earth. At this point, another group of organisms evolved that could manufacture their own organic nutrients. But how did they do this? They used simple inorganic molecules such as carbon dioxide and hydrogen sulfide that were present in Earth's atmosphere for manufacturing their own food. These organisms were called autotrophs, meaning self-feeders. Over the course of evolution, based on the source of energy utilized, two main types of autotrophs evolved. One group was chemoautotrophs, which used chemical energy to prepare their food, and another group, photoautotrophs, which used sunlight to prepare their food. The process by which the second group of autotrophs prepared food using sunlight is called photosynthesis. In photosynthesis, energy from sunlight is transformed into chemical energy that is stored in carbohydrates and other organic molecules. Without this flow of energy from the sun, the life on Earth will eventually come to an end. We just learned that the first photoautotrophs, for example green sulfur bacteria and purple bacteria, used hydrogen sulfide and carbon dioxide for preparing food. This may sound strange for we human beings because an atmosphere of hydrogen sulfide is extremely hostile for us. This gas has a characteristic foul smell of rotten eggs. Also, it is very poisonous and corrosive. But primitive photoautotrophs used hydrogen sulfide as their source of electrons for photosynthesis, and the overall reaction is CO2 plus 2H2S gives CH2O plus H2O plus 2S. Here CH2O represents a unit of carbohydrate because 6 times CH2O equals C6H12O6, which is a glucose molecule. Glucose is the simplest useful form of carbohydrate produced during photosynthesis. See, in this photosynthesis, the products are carbohydrate, water and sulfur. It does not produce oxygen, so it is called anoxygenic photosynthesis. Now it is the time of the second famine. Hydrogen sulfide in the Earth's atmosphere started to get depleted, and at this point, a new photosynthetic prokaryote called the cyanobacteria came to Earth. Cyanobacteria can utilize water, which was much more abundant than hydrogen sulfide at that time. The overall reaction for that process is CO2 plus H2O gives CH2O plus O2. This reaction produced a precious waste product, oxygen. It was precious not for the cyanobacteria but for many other life forms on Earth. This kind of photosynthesis, which produces oxygen as a byproduct, is called oxygenic photosynthesis. But where did the oxygen come from? From carbon dioxide or water? Scientists once thought that oxygen was released from the carbon dioxide molecule. But in 1931, C.B. Van Niel proposed that oxygen comes from water and not from carbon dioxide. He had a reason for that. Look at these two photosynthetic reactions. The first one utilizes hydrogen sulfide, H2S, and the second one utilizes water, H2O. In fact, H2S and H2O are analogous because, as you know, sulfur and oxygen belong to the same group in the periodic table. So Van Niel thought that just like sulfur originates from H2S in the first reaction, oxygen originates from H2O in the second reaction. Remember that he made this statement when he was a graduate student of Stanford University. And the scientists were not convinced about this. 10 years later, in 1941, Samuel Rubin and Martin Kamen used a heavy isotope of oxygen, O18, to trace the oxygen and prove that Van Niel was right. And how did they prove it? They took cultures of green algae chlorella in two different containers. The first culture was given O18 labeled carbon dioxide and normal water, whereas the second culture was given normal carbon dioxide and O18 labeled water. The oxygen released from the first culture was the normal oxygen, whereas the oxygen released from the second culture was O18 labeled oxygen. So what do we learn from this experiment? Water is the source of oxygen produced by photosynthesis. O18 is not radioactive, so they used mass spectrometry to quantify O18. The complete balanced equation for photosynthesis now becomes 6CO2 plus 12H2O gives C6H12O6, which is a glucose molecule, plus 6O2 plus 6H2O. Here it means that one glucose molecule is produced from 6 molecules of carbon dioxide. Now let's see the sites of photosynthesis. In 1881, a German biologist, T.W. Engelmann, identified chloroplasts as the sites of photosynthesis. Most chloroplasts are seen in the mesophyll cells of leaves. If you take a cross-section of a small part of this leaf and examine under a light microscope, you can see the leaf anatomy like this. Here, below the upper epidermis, we can see vertically elongated palisade parenchyma cells. Below the palisade tissue layer are the layers of oval-shaped spongy parenchyma cells. Palisade and spongy parenchyma cells together form the mesophyll tissue in leaves. Even though both palisade and spongy parenchyma cells contain chloroplasts, palisade parenchyma cells contain more chloroplasts than spongy parenchyma. Most of the photosynthesis takes place in the palisade cells. Now if you go for a higher magnification of a single palisade cell, you may find about 40 to 50 chloroplasts. If we want to see the ultrastructure of a single chloroplast, then we have to use an electron microscope. The transmission electron micrograph of a single chloroplast obtained using a transmission electron microscope is shown here. Now, let's learn about the structure of chloroplasts in detail. They are disc-shaped. They are surrounded by a double membrane called the chloroplast envelope. The outer membrane contains porins and is permeable to metabolites of small molecular weight. But the inner membrane of the chloroplast is highly specialized with transport proteins. These transport proteins regulate the movement of metabolites into and out of chloroplast. In other words, the inner membrane is selectively permeable. The outer and inner membranes of chloroplast don't contain chlorophyll. The two envelope membranes are separated by a gap called the intermembrane space. There is a colorless fluid within the chloroplast called stroma. Stroma contains the enzymes responsible for carbohydrate synthesis. In addition to the inner and outer membranes of the envelope, chloroplasts have a third membrane system called the thylakoid membrane. This membrane forms a network of flattened sacs called thylakoids. The word thylakoid is derived from the Greek word thylakos, which means sac. Thylakoids are arranged in orderly stacks called grana, which resemble a stack of coins. A single stack of thylakoids is called granum. Each granum contains around 10 to 20 thylakoids. These are also called granule thylakoids. There is another type of thylakoids called stromal thylakoids. They join the granum stacks together and make the thylakoid membrane network a single compartment. Stromal thylakoids are also called stromal lamellae or frets. The space within the thylakoids is called the thylakoid lumen. Thylakoid lumen is filled with a fluid. Okay, now where are the photosynthetic pigments located? Chlorophyll and other pigment molecules are embedded in the thylakoid membranes. They are organized in special units called photosystems. Each photosystem is an assembly of 250 to 400 pigment molecules. There are two different types of photosystems, photosystem 1 and photosystem 2. We will learn more about photosystems later. In the stroma, you can also see strands of DNA and prokaryotic-like ribosomes, that is 70S ribosomes. Chloroplasts are cell organelles, right? Normally, we don't expect DNA to be present in a cell organelle. Have you ever wondered why chloroplasts have their own DNA and prokaryotic 70S ribosomes? According to the endosymbiosis theory, chloroplasts were once photosynthetic bacteria. Bacteria are prokaryotes. And they got engulfed by a large eukaryotic host cell. Then the photosynthetic bacteria and the eukaryotic cells started a symbiotic relationship with each other. The photosynthetic prokaryotic bacteria provided important nutrients to the eukaryotic host cell, and in turn these photosynthetic bacteria received protection and a steady environment to live in. Since chloroplasts were once photosynthetic bacteria, they have their own DNA and prokaryotic 70S ribosomes. Chloroplasts often contain starch grains and small lipid droplets, which are the reserve food materials. Now, an interesting fact about chloroplasts. If you can view a live mesophyll cell, you can see that when sunlight hits the leaves, chloroplasts change their positions. Yes, the chloroplasts move inside the mesophyll cells in order to optimize their activity, as you can see in this video. They move towards weak light to perceive light efficiently, whereas they escape from strong light to avoid photo damage. To watch the full video, check out the link in the description box below. Now let's see the pigments which play an important role in photosynthesis. As we saw earlier, there are photosystems embedded within the thylakoid membranes. Each photosystem has a light harvesting complex. The light harvesting complex, also called as antenna complex, consists of proteins and pigment molecules. The light harvesting complexes capture light energy and increase the efficiency of photosynthesis. We will learn more about them later in this video. A pigment is any substance that absorbs visible light. The pigments that help in absorption of light during photosynthesis belong to three main groups: the chlorophylls, carotenoids, and phycobilins. The main photosynthetic pigments are the chlorophylls. They are greenish pigments. Different types of chlorophyll pigments are chlorophyll A, B, C, D, E, F, and bacterial chlorophyll. However, all these chlorophylls are not present in all photosynthetic organisms. Chlorophyll A is the most abundant type of chlorophyll. It occurs in all photosynthetic eukaryotes and in the prokaryotic cyanobacteria. Chlorophyll E and F are the rare forms of chlorophyll found only in a very few organisms. As chlorophyll A is the most abundant and important chlorophyll, let's learn about its structure. Chlorophyll A is a large molecule with a central core of magnesium held in a porphyrin ring. Attached to the ring is a long carbon-hydrogen chain. It helps to anchor the chlorophyll A molecule in the thylakoid membranes of the chloroplast. The second group of photosynthetic pigments are the carotenoids. They are usually red, orange or yellow pigments. Two groups of carotenoids, that is carotenes and xanthophylls, are usually present in all chloroplasts. Carotenes are typically orange and yellow pigments, while xanthophylls are yellow pigments. The carotenoids also act as an antioxidant to prevent photooxidative damage of chlorophyll molecules. Do you know what causes the bright red color in tomatoes? It's lycopene, a kind of carotene. Carotenes were first discovered in the early 19th century in carrot roots, and so the name carotenes. Let's learn about the structure of beta-carotene, an important carotene. Beta-carotene is made up of 8 isoprene units, which are cyclized at each end. The long conjugated chain is responsible for the orange color of beta-carotene. Conjugation? What's that? Conjugation means that the single bonds and double bonds are arranged in an alternate fashion. As you can see, there are 11 double bonds in alternate positions in the structure of beta-carotene. As the conjugation increases, means as the number of double bonds increases, the energy difference between the HOMO and LUMO of the molecule decreases. HOMO and LUMO. Does it sound complicated for we biologists? In fact, it's very simple. Let me explain. In a molecule, the electrons are sitting in molecular orbitals. There are occupied orbitals and unoccupied orbitals. The levels here indicate energy of the orbitals. Among the occupied orbitals, the one with highest energy is called HOMO. HOMO means the highest occupied molecular orbital. Among the unoccupied orbitals, the orbital having the least energy is called LUMO. LUMO means lowest unoccupied molecular orbital. If enough energy is supplied to the electrons in HOMO, they can jump to LUMO. So let's come back to the condition when conjugation increases in a molecule. That is, when HOMO and LUMO gap is very less. Radiation of low energy is required for the transition, and this radiation falls in the visible region of the electromagnetic spectrum. This is why the molecule exhibits color. The color that you see depends on the exact energy difference of HOMO and LUMO. So now imagine what happens if the conjugation is very less, or if there is no conjugation at all. Then the energy gap between HOMO and LUMO will be very high. As a result, an electronic transition between these levels requires high energy, and such an energy can be contributed only by UV radiation and not by the visible light. So such molecules appear colorless because UV light is not sensible to our eyes. Okay, now let's move on to the third group of pigments, the phycobilins. The word phyco refers to algae. Phycobilins are seen in the chloroplast of cyanobacteria and red algae. Within the group of phycobilins, phycocyanin and phycoerythrin are of importance. Phycocyanin is a light blue pigment and phycoerythrin is a red pigment. You may remember that chlorophylls and carotenoids are found in the photosystems embedded in the thylakoid membranes because they are fat soluble. Unlike chlorophylls and carotenoids, phycobilins are water soluble pigments. They form protein complexes called phycobilisomes on the surface of the thylakoids rather than being embedded in the thylakoid membranes. So that's all about the photosynthetic pigments. Actually, the reason why there are so many photosynthetic pigments is that each pigment absorbs light more efficiently in different parts of the electromagnetic spectrum. The absorption pattern of a pigment is known as its absorption spectrum. Chlorophyll A from the group chlorophylls absorbs well at a wavelength of about 400 to 450 nanometer and also at 650 to 700 nanometer. Chlorophyll B absorbs well at 450 to 500 nanometer and also at 600 to 650 nanometer. Carotenoids absorb well at 400 to 530 nanometer. This means that the absorption spectra of these pigments are non-overlapping, and this broadens the range of light that can be absorbed in photosynthesis. None of the pigments absorbs well in the green-yellow region, which means that light in the green-yellow region is reflected the most, and this is why plants appear to be green. Have you heard of action spectrum? An action spectrum is the graphic representation of the effectiveness of different wavelengths of light in photosynthesis. It is a plot showing relative efficiency of photosynthesis produced by light of different wavelengths. In the action spectrum, we can see that the maximum photosynthesis occurs in blue and red light, and the minimum photosynthesis occurs in, of course, green light. Let's come back to the photosystems. In each photosystem, there is a reaction center. A reaction center consists of a complex of several proteins, chlorophyll A molecules, a primary electron acceptor and other cofactors that together execute the primary energy conversion reactions of photosynthesis. Chlorophyll A is known as the core pigment. There are 6 chlorophyll A molecules in the reaction center and they are called reaction center chlorophylls. Out of this, 2 of them form the special pair chlorophylls. They are called special pair because only this special pair of chlorophyll A molecule can take part in the primary photochemical reaction in photosynthesis. All other photosynthetic pigments in the light harvesting complex are called antenna pigments or accessory pigments. They consist of mainly chlorophyll B and carotenoids. Some chlorophyll A molecules will also be present in the light harvesting complex. The antenna pigments are named so because they form an antenna-like network for the gathering of light. Almost all the light energy absorbed by accessory pigments is transferred to the special pair of chlorophyll A molecule in the reaction center by the resonance. This transfer is also known as Forster transfer. So in this video, we learned how photosynthesis and photosynthetic organisms evolved on Earth, also about 2 different kinds of photosynthesis, the detailed structure of chloroplast, the location of photosynthetic pigments, and how the antenna pigments transfer the light energy to the special pair chlorophyll A molecule in the reaction center of each photosystem. Now, the special pair chlorophyll A molecule is ready to take part in the primary photochemical reaction of photosynthesis. In the part 2 of this lecture, we will see the primary photochemical reaction in detail. Thank you for being with me and stay curious.